everyone, welcome back to my channel. Today, we're diving into the fascinating world of bioinformatics file formats. Now, you might be wondering, why should I care about file formats in biology? Well, my friend, let me tell you, this is one of the most crucial aspects of modern research and data analysis in the biological sciences. So, buckle up and get ready to have your mind blown. In this playlist, we'll be delving into conversations surrounding the VCF and gVCF file formats. If you haven't already subscribed to my channel, I encourage you to do so for the most up-to-date videos and solutions related to bioinformatics. The variant calling format VCF file serves as the output in a bioinformatics pipeline, acting as a textual repository for gene sequence variations. In a standard variant calling workflow, a DNA sample is subjected to sequencing through a next-generation sequencing system, yielding raw reads formatted as FASTA or FASTQ files. These raw reads undergo alignment to produce BAM or SAM files, as detailed in previous videos. If you are unfamiliar with these file formats or seek insights into their data organization, I recommend reviewing the linked videos in the description. Utilizing the SAM or BAM file, a variant calling algorithm or tool discerns alterations in a specific genome relative to the reference genome. These variations are then documented within a variant calling file, a VCF. The VCF captures diverse sequence variations encompassing SNPs, insertions, deletions, and structural variants. Given the potential enormity of some VCF files, loading the entire file into memory becomes impractical. Consequently, strategies such as employing Linux or Unix tools to manipulate or selectively access specific segments of the file are implemented. A critical cautionary note emphasizes that a VCF file, being a text file, must never be opened in a text editor like Microsoft Word, as this could disrupt formatting and potentially harm the file. Instead, it is recommended to open such files within a Linux or Unix environment, ensuring a secure examination, exploration, or extraction of pertinent information, either in its entirety or selectively by retrieving specific lines. Let's go through with detailed version of VCF file. Upon opening a VCF file, the initial view typically resembles the following. The first few lines constitute the introduction to the VCF file. A VCF file, in essence, is organized into three distinct sections. The foremost section comprises metadata lines, constituting the initial lines of the file. These lines, denoted by a prefix of 2-pound symbols, serve as the preamble to the VCF file and encapsulate critical information about the file's format and content. Following this, the next section is the header section, represented by a single header line prefixed with a pound symbol. Subsequently, the file transitions to records, constituting the data lines. These data lines contain various details about genetic variants, and the subsequent slides will delve into the specifics of each section. Initiating with the metadata lines positioned at the file's outset. These lines delineate the format and specifics of the particular VCF file. The information encapsulated in these lines may encompass details about the sequencing process, variant calling software, or the reference genome employed for variant determination. As reiterated earlier, these lines, characteristic of metadata, commence with a prefix of 2-pound symbols, signifying their distinct role in outlining crucial file characteristics. The initial line in this section designates the file format, explicitly indicating the format of the file. Following this, there are several additional lines containing diverse information. Subsequently, a set of lines emerges, commencing with info, followed by another set starting with filter, and further lines initiated with format. Specifically, the lines beginning with info provide descriptions of annotations intended to characterize variant sites within our dataset. For instance, within the data lines corresponding to each variant site, details encompass the number of samples with data, total depth, allele frequency, and more. The metadata lines prefaced by info furnish the descriptions and anticipated values for these annotations. Moving on, lines starting with filter outline the filters applied to our dataset. Lastly, lines beginning with format mirror the structure of info lines. To elucidate, when detailing a sample, information within the data lines encompasses annotations such as genotype quality, read depth, and related information, each assigned a specific value. Descriptions of these annotations and the expected values are elucidated in the metadata section through lines prefaced by format. If the distinction between lines starting with info, filter, and format is not immediately apparent, clarity is expected to emerge as we progress to discussing the records in the VCF file. This subsequent discussion will expound upon the details regarding variants and the information pertinent to both variants and samples. During this phase, numerous annotations will be addressed, enhancing comprehension of the delineated distinction. Now, delving into the subsequent section, which commences with a header line, it's imperative to note that the first 8 columns in the ensuing section are obligatory. These columns, tab-delimited, encapsulate properties observed at the variant level. In instances where a VCF file accommodates multiple samples, certain site-level information will be an average or summary of values obtained for that site from various samples. Proceeding further, we can acquire sample-specific information, such as genotype or genotype quality, in a column-termed format, positioned as the 9th column. Following this, individual columns, one for each sample, extend beyond column 10, providing annotation values for each sample in a distinct column. The subsequent lines following the header line constitute the record lines. Essentially, these lines act as repositories, storing comprehensive information about variants and details concerning both the variants and the respective samples. Now, let's delve into the specifics of each of these columns to comprehend the stored information. The initial column signifies the chromosome and holds data about which chromosome the variant was detected. The subsequent column denotes the position, specifying the variant's location on that chromosome. For multi-base variants, the position corresponds to the first base's position, while for deletion events, it corresponds to the base preceding the event, i.e., before the deletion. The I column serves the purpose of indicating whether the variant has been previously classified and is present in any database like DBSNP. Depending on the variant, its position on the chromosome, and whether it's previously classified or part of a database, the corresponding identifier is displayed. A .ASCII indicates that the variant has not been previously classified. Moving on, the reference column delineates the base present in the reference genome at that position in that chromosome. Conversely, the alternate column indicates the base present in our dataset at that specific position. The alt column accommodates variants found in our dataset. If there are multiple variants present, they will be separated by commas. For events like insertions, the alt column will additionally include the bases preceding the insertions. This inclusion allows understanding the insertion's position in comparison to the reference sequence. It's important to note that the bases reported in the ref and alt columns are consistently provided on the forward strand. Moving on to the subsequent columns after reference and alternate LA, we encounter the quality column. The values in this column represent probability values estimating whether the variant call at that site results from sequencing error. For instance, a value of 10 suggests a 1 in 10 chance that the called base is not a variant but rather a result of sequencing error. Higher quality values signify greater confidence that the base is genuinely a variant and not an erroneously called base due to sequencing error. The subsequent column is labeled filter, which corresponds to filters applied to our dataset. To ascertain the applied filters, one should refer back to the metadata section and inspect the lines beginning with filter. In the provided example, 2 filters were applied, including one to identify variants with a quality score less than 10. If no filtration is applied to the records, the filter column will exhibit dots in lieu of pass or filter names. Following the filter column, we encounter the info column offering information on specific abbreviations for these sites. To comprehend these abbreviations, one can refer to the metadata section and explore the lines starting with info. Here, descriptions and names of abbreviations are provided. Examples of such abbreviations include Ns and number of samples with data, DP unfiltered combined depth across all samples, and AF allele frequency for alternate alleles. Additional abbreviations and their descriptions can be extensively found on forums like Get, providing comprehensive documentation on these abbreviations, their expected values, and how to interpret them. Lastly, transitioning to the sample-level annotations and focusing on the format column. This column contains a colon-separated list of abbreviated categories describing these samples. To comprehend the meaning of each abbreviation, one can refer to the metadata section, specifically the line starting with format, to gain insights into what these abbreviations represent and the values they are expected to have. The list separated by colons typically initiates with GT representing genotype. Subsequently, we find genotype followed by genotype quality depth and haplotype quality. As an illustration, consider the last entry in the format column. For this entry, the format column includes genotype quality, and depth. The data for the subsequent samples adheres to the same sequence. To elucidate further, examining the last record in the format column reveals that the genotype for all 3 samples follows a consistent order. For instance, the genotype for sample 1 is 1-1, while for sample 2, the genotype is 0-0. Following this, we encounter genotype quality with a value of 12 for sample 1 and 20 for sample 2. Lastly, the depth for sample 1 is 3, while the depth for sample 2 is unavailable. Genomic VCF file, a genomic variant calling format ECF file, commonly known as a genomic VCF file, serves as a standard in bioinformatics for representing genetic variations across a genome. This file captures information about various variants, encompassing single nucleotide polymorphisms (MPs), insertions, deletions, and structural variations identified through the analysis of DNA sequencing data. Each entry in the file corresponds to a specific genomic position, providing a comprehensive account of observed variations along with pertinent annotations. In contrast to VCF files, which exclusively record variant sites, genomic VCFs document every position in the genome, whether variant or non-variant. These genomic VCFs are generated using the GAT program called haplotype caller, particularly in GAT versions 3 and above. 2 types of genomic VCFs can be produced, one featuring individual records for every site, regardless of its variant status, and the other containing individual variant records followed by grouped non-variant sites, forming a non-variant block of records. Genomic VCF files play a pivotal role in genomics research, facilitating the identification and characterization of genetic variants across diverse individuals or populations. These files find application in various contexts, including variant discovery, association studies, and the interpretation of genomic variations in relation to diseases or traits. That concludes the content for this video. I trust you found this video to be informative. If it provided value, kindly consider subscribing, liking the video, sharing it, and leaving your comments in the comment section. Until our next encounter, take care and see you soon.